Hi everyone, my name is Rashmi Dhital and I'm an instructor in the Division of Rheumatology at Vanderbilt University. Today I'll discuss the results of our work on human milk fatty acid profiles among breastfeeding mothers with and without systemic autoimmune rheumatic diseases. Systemic autoimmune rheumatic diseases, which I'll refer to as SHARDs, are characterized by chronic systemic inflammation and commonly require immunomodulatory treatments. Some rheumatic diseases, such as systemic lupus erythematosus, SLE, and rheumatoid arthritis, RA, disproportionately affect women and are commonly diagnosed among women of reproductive age and thus may have pregnancy and breastfeeding implications. Such diseases, as well as therapies used to treat them, may affect lipid metabolism and influence breast milk composition. However, data are limited. So our objective was to characterize and compare fatty acid profiles in breast milk of mothers with and without SHARDs. And because nearly all long-chain fatty acids in breast milk are derived from the circulation by lipoprotein lipase, we hypothesized that chronic inflammation in SHARDs may suppress lipoprotein lipase, reducing mammary uptake of long-chain fatty acids more than 16 carbons, with resultant lower concentration of long-chain fatty acid in milk from SHARD patients. We analyzed archived breast milk samples from 44 breastfeeding women enrolled in the Human Milk Research Biorepository at UCSD. 29 had SHARDs, of whom 15 had rheumatoid arthritis, 11 had lupus, and 3 had both. They were matched to 50 healthy controls on maternal and infant ACE. Fatty acid concentration was assessed at the UCSD lipidomic score using liquid chromatography mass spectrometry. We compared baseline demographic and clinical characteristics between SHARDs and controls using t-test for continuous and Fisher's exact test for categorical variables. We then compared median fatty acid levels between the two groups using Wilcoxon rank sum test. In a subgroup analysis, we assessed fatty acid levels separately for RA and SLE. Then, to assess the association between SHARD status and fatty acid level, we first log transformed the fatty acid concentrations to approximate normality, followed by multivariable linear regression models, adjusting sequentially for maternal, infant, and sample-related covariates, as shown in the blue font below. In Model A, we adjusted for various maternal and infant covariates, as well as milk collection characteristics. The Model B adjusted for variables in Model A, as well as storage length of the milk samples. And the final Model C adjusted for SHARD therapies, such as corticosteroids and disease-modifying anti-rheumatic drugs, in addition to the other variables. In this table, comparing baseline maternal characteristics between SHARDs and controls, we observed that maternal characteristics were comparable between groups. Similarly, obstetric and infant characteristics were similar between SHARDs and controls, except for slightly earlier gestational age at delivery among SHARDs, 38.4 weeks versus 39.72 weeks in controls. In terms of milk storage characteristics, we did note that the average storage duration of milk samples for our SHARD patients was considerably longer, 40 months compared to 3 months in the control group. And this was something that was inadvertent and something that we figured out during data analysis, so after the fact. In terms of exposures, as expected, the use of DMARDs or disease-modifying anti-rheumatic drugs was exclusive to the SHARD groups, and corticosteroid use was more common in SHARDs than controls. Total milk macronutrients in terms of total carbohydrates, lipids, and proteins were similar between SHARDs and controls. Now, to discuss the results of our fatty acid analysis, we observed that the levels of various individual medium and long-chain fatty acids were higher in milk samples from SHARDs compared to controls. Specifically, the ones highlighted in orange here, lauric acid, myristic acid, palmitic acid, and oleic acid were significantly higher in the SHARD group compared to controls. All the other fatty acids shown in the tables also tended to be higher in the SHARD group, but did not reach significance. In this table, we see the fatty acids that tended to be lower in the shard group, and cirrhotic acid, which is a very long chain saturated fatty acid, was significantly lower in shards. In terms of the most abundant fatty acid levels in both these groups, the following 5 fatty acids were most common, and they were oleic acid, linoleic acid, palmitic acid, lauric acid, and myristic acid. This subgroup analysis shows differences in fatty acid in SLE and RA separately compared to controls. The blue line here represents SLE, the green line is RA, and the orange is controls. And we noted that the difference in fatty acid between shards and controls was primarily driven by SLE. And so SLE, but not RA compared to controls, had significantly higher levels of several fatty acids. This forest plot here shows results of our multivariable linear regression analysis to study association between shard status and fatty acid levels. And we observed that shard remained persistently associated with higher fatty acid levels despite adjusting for covariables. And in general, the magnitude of association became stronger after sequential modal adjustments for storage length and shard therapies. For example, the first fatty acid shown here, lauric acid, was associated with 1.4 to 2-fold higher values in shard patients, with the highest estimate for the fully adjusted model, which included storage length and shard therapies. Conversely, cirrhotic acid was significantly lower in the base model and model A, but the difference was attenuated after adjusting for storage, duration, and shard therapies. Because we noticed that the storage length for our study was significantly different between shards and controls, we studied fatty acid levels based on storage duration of the milk samples. The first figure here is for controls, and we can see that all the samples were stored for less than a year before analyzing, whereas in the shard group, 16 out of the 29 patients had a storage duration of more than 36 months. And although the differences in fatty acid levels for shards across storage duration did not reach statistical significance, we noted that most fatty acids showed higher levels at intermediate storage duration between 12 to 24 months. And a possible explanation is that endogenous lipases in milk gradually break down more complex lipids over time, resulting in release of free fatty acids. So to conclude, despite comparable level of macronutrients, we noted variability in fatty acids in milk from shard patients compared to controls. The most abundant fatty acids were similar in both groups, suggesting a consistent profile in human milk independent of autoimmune disease status. The elevated levels of various medium and long-chain fatty acids observed in moms with shard, particularly those with lupus, may imply possible influences of inflammation as well as shard treatment on milk composition. For example, studies have shown differences in fatty acid profiles based on local inflammation such as mastitis, and a study also showed that higher cortisol in human milk was associated with higher levels of lauric acid and myristic acid. And so it is possible that shard therapies influence fatty acid environment. However, our data also showed that storage time was associated with higher fatty acid levels, which could have led to higher levels in our shard patients. Given this, future research matching controls based on storage duration will help improve data interpretability, reduce bias, and help explore clinical significance of fatty acid compositional differences. And so learning from this for our proposed project to study macronutrient concentration in the overall shard samples in the human milk research biorepository, as well as to study micronutrients and human milk oligosaccharides in a subset of samples. We did match by storage duration in addition to maternal A's and child A's. And so with that, I am happy to discuss and to get any feedback on the project. Thank you for listening.